The art and science of compost, maximizing the soil food web for your farm and garden ecosystem. I put that little quote there, composting, because I know on your program it says the art and science of compost, but as I was putting this together, I realized that part of the point of this talk is about defining compost as a verb rather than a noun. Because as we get into it, you start to learn about the reality of compost. It's a process. It's never really done. It's not a thing. Most of you don't know me. I'm from Jersey, born and raised in Middlesex County in Denellen. I spent the last handful of years in different states working and living on organic CSAs and other farms. Got my permaculture design certification out in Fairfield, Iowa. That's where I studied sustainable living. Got to spend a month working and living with Elaine Ingham, studying the soil food web and composting. That's where this stuff really came alive for me and where I started to really dive deep. And as a result of that, I came to co-found the Living Soil Lab, which was out in Fairfield. We were doing microscopy for the community, offering soil fertility services, building compost, brewing extracts and tea, doing soil samples, and really testing this stuff out. And that's where I got to see some of how this really works. Since then, I've gotten to have some experience advising some different projects along the lines of the soil food web and soil fertility. So most of you, right, all of you don't know me at all, so it's nice to meet you, and thanks for coming out. By the time we're done, probably an hour and 15, hour, 20 minutes, I hope that you'll have an inspired understanding of the soil food web. It's not going to be a straightforward talk on compost. We're going to go deep in a short amount of time. We're going to cover a lot of ground, and hopefully by the end of it, you'll also have an ability to think critically about making compost tailored to fit the specific plant range you want to thrive. Also, I hope you'll leave empowered about the diversity of options for executing these concepts in ways that fit your practical parameters. No one-size-fits-all solution for composting, as most of you already know, and I'm not going to be putting it hard and fast. Hopefully you'll be able to work with it and make it work for you. I hope by the time we're done, you'll also have a sense of enchantment for the great mystery of our interconnectedness. Because ultimately, the story of the soil food web is a story about interconnectedness, and it's a story about complex systems dynamics, coevolution, intelligent, self-organizing systems, and how people, plants, microbes, the whole gamut play a part in the whole shebang. So, basic components of the soil food web. This is a chart from the USDA Soil Biology Primer. Sun, water, air, sand, silt, and clay, dirt, plants and their roots, and then the invisibles, the bacteria, fungi, protozoa, nematodes, microarthropods, and then the bigger creepers and crawlers, the earthworms, and then the moles and the birds, and on up to the animals and people. We have an impact on the soil food web, but mostly during this session we're going to focus on the invisibles and their relationship with the plants. More likely than not, the soil food web is a shell of itself. In your soil, on your land, wherever you are, to varying degrees, at varying times. I mean, depends on the history of your land. Was it tilled historically every year? Chemicals applied in the past? Is it an urban lot? Is it reclaimed from building zones? How compacted is that soil? All the things that happen over time have adversely affected the diversity of these organisms. And most of the soils that I've sampled and looked under the microscope at 400 meg, you can see these guys running around. It's just mostly bacteria. The diversity of the web, the good guy fungi, the good guy nematodes, the protozoa, the full spectrum that's responsible for cycling nutrients and building soil, it's not there. Even in forest soil, it's not always there. And it's a result of logging, ecosystem disturbance, disturbs the life in the soil. So I'll be talking about compost as a means to restore the health of the soil food web and the diversity of the soil food web. But first I'm going to be giving a basic overview of the benefits of a good, healthy soil food web. Here's a microscopic image of fungal hyphae. They're one cell wall thick microorganisms that are invisible. They run underground. Each hyphae extends out from the general body, making up the body of the fungus. And the largest organism on Earth, anybody know? Paul Stamets. Discovered, yep, up in Washington State. I think the body of this mycelial network is about 20 miles. And they're secreting enzymes and acids that break down organic matter to absorb directly into their bodies. So they're moving and transporting nutrients throughout these networks. And some of these fungi are root mutualists, so they live in and around root systems and transport water and nutrients throughout the different root zones and the rhizospheres of these plants. That little gold nugget there is a humus particle full of delicious nutrients. And you may or may not be able to see the bacteria. It's not very clear here. That might be one. But they're there too. They're tiny. So this is the mycelial network. So they're second, tertiary level organisms. They're primary and secondary decomposers, bacteria and fungi. Not all bacteria and fungi are decomposers. Some cycle nutrients, nitrifying bacteria, nitrogen-fixing bacteria. There's also the parasitic and pathogenic organisms. We're not so interested in them as we are with the beneficials. And this could be a talk in and of itself, talking about these microbes and what they do. I refer to my paper if you want to learn a little bit more about them. I have that up online. I'll give you guys the links later, along with links to go deeper if you want to learn more. But they're breaking down organic matter. They're pulling together particles in the soil, building microaggregates, building air structure, pore structure in the soil, and they're enabling deeper root penetration, more aeration in the soil, deeper water channels for your plants to grow fuller, more robust, and gain more access to nutrients in the soil. In essence, when you think about bacteria and fungi, think about them as bags of fertilizer. And then these other guys, they're the fertilizer spreaders. The nematodes on the bottom. Protozoa, that's the ciliate. Microarthropods. They're not only shredding and working with the organic material, making it more available for bacteria and fungi to get to, but they're also eating bacteria and fungi all day, every day, releasing plant-available nutrients in their wake, and leaving air and pore space through the tunnels that they create. These guys are also taxicabs for bacteria, and one of the main ways that bacteria gain mobility through the soil to spread their benefits. So, nematodes tend to get a bad rap, mainly because farmers tend to know them as root-feeding nematodes. Root feeders are the smallest organisms of the nematode species, and they tend to thrive in compacted soil, anaerobic soil, where these larger predatory and bacterial and fungal-feeding nematodes can't really thrive. So there's not enough room in the soil, their populations are going to be low, root feeders are going to thrive. So think about nematodes along the spectrum. We want fungal feeding, bacterial feeding, beneficial predatory nematodes. And you can reintroduce them through good compost. The stuff that you buy in the inoculants, Lane was talking about this yesterday, it's more geared toward pests, combating pests, but the ones that are going to be eating bacteria and fungi and cycling good nutrients, you gotta get them through the compost. And they reproduce. They'll have 200 children, 40,000 grandchildren. Once you get them in the soil, they're going to go to town, and you're going to love what they do. Similar to earthworms. Protozoa, ciliate is one. flagellates, amoeba, and testate amoeba are other types of protozoa. This is a picture of a protozoa that you don't want to see. Ciliates are eating bacteria and fungi, but they also thrive in anaerobic soils. So where you have a predominance of ciliate in your compost, in your soil, your plants aren't going to be very happy there. So you'd rather see flagellates, you'd rather see testate amoeba. When you look at these guys under the microscope, you'll be able to tell to some degree the quality of the soil based on the balance of microorganisms that you have there. Microarthropods, again, shredders, nutrient cyclers, fertilizer spreaders. That's what these guys do. So here's a picture of aerobic healthy soil. Scanning electron micrograph. One thing you'll notice is all the shadows. These are the pore spaces. These are the air spaces. This is what your organisms are doing. There's room for roots, there's room for air, there's room for water to penetrate, and there's room for the organisms to move around. And that's what they're creating when you have them in abundance. On the other hand, it's an anaerobic putrid soil sample they took. And the only airspace here are cracks. There's not much more than bacteria that can really work here. So, and in a lot of ways, this is what a lot of compacted soils look like. Roots aren't doing very well here. The rest of the organisms aren't doing very well here, and that's why they're not there. So they not only thrive in good soil structure, but they build good soil structure. And we're going to talk about the nutrient cycle a little bit and how these guys impact the nutrient cycle. So soil organisms are the X factor for growing healthy plants. This is what I'm getting at. And compost is your best tool for bringing them back into the farming garden. Okay. Now before I go any further, because now it's early, I just want to pause. Because we're diving right in. But I just want to pause and ask you guys to close your eyes. Close your eyes. Take a deep breath. And see your farm or garden as it is now. See all the plants, the animals there. Who else lives there? What's the quality of life like there? Zoom out and see the land as a whole. How does it feel? Pressure, worry, anxiety, fulfillment, excitement. Now, see the farm or garden of your dreams. Is it the same land? Is it a different land? What type of plants are growing here? How are they doing? Animals? Who do you live with? What's the quality of life like here? What's the community like that you're embedded in? Feel the context. And what's your impact on the community? Alright, now take another deep breath, open your eyes. So, I just want you to keep the big picture in mind as we go through this. So as you guys know, we were there for 6 hours. There's a lot of details. There's a lot of details here. And I just want to see if we can keep this feeling in the room. Okay. So soil organisms are the X factor for creating healthy soil and thus creating healthy plants. Good farmers and gardeners know it's wise to utilize practices that in the long run, increase the percentage of soil organic matter and humus buildup in the soil. That's not news. But as you know, there's varying levels of organic matter in the soil. You might have 2%, you might have 10%. But whatever percentage of organic matter you have total in your soil, it varies. But what makes up that percentage of soil organic matter is relatively consistent. 5% of your total soil organic matter is plant residue, less than 5%. That's not a lot. The organic matter that you put down, the plant stuff that you leave on your fields, the mulch that you put down, less than 5% of soil organic matter. The other 95% is the direct or indirect result of soil organisms. About twice as much of the soil organic matter is actually comprised of the bodies of these organisms, the carbon-based shells and structure of the bodies of the organisms in your soil. And then 33 to 50% be decomposing organic matter. Organisms, of course, are responsible for that. And the other 33 to 55 or 50% is already decomposed organic matter, the humus. Does that make sense? So whatever percentage soil organic matter you have, good, we want more of it. How do you get more of it? You need the organisms. The more you have, the quicker the buildup of soil organic matter and humus, the more fertile and rich your soil is going to be. And your plants are going to like you for it. So, before we get there, most of this activity goes on in the top few inches of the soil, where the fresh plant residue meets the soil surface. That's where a lot of this microbial activity is going on. But really, where it's really going on is in the rhizosphere, in the zone right around the root system of the plants, about a millimeter on either side of the root hair. This study looked at 2 different soil samples and compared the total bacteria and fungi in the rhizosphere as compared to the surrounding soil. In both cases, they found about twice as much activity, bacterial activity in the rhizosphere of the plant as opposed to the surrounding soil. About 4 times as much fungal activity in the rhizosphere as in the surrounding soil. Okay, why? Root exudates. This is, you guys recognize this image from yesterday. Okay, so 75 to 80% of the plant's energy is going into its root systems. It varies plant to plant, but the vegetable plants, the stuff we're growing, row crops, berry bushes, orchard trees, 75 to 80% of their energy is being pushed down into the soil, into their roots. So I'm a plant, right? I'm doing what I do. Taking in the energy from the sun, the carbon from the atmosphere, making simple sugars. Making carbon chains, the basis of all life. That's what plants do that are so special. And that's what these organisms can't do, and that's what they need. So of that 75 to 80% of sugars, carbon chains, cakes, and cookies, is going down into the root systems. Half of that is leaving the root systems. That's what root exudates are. They also release those cakes and cookies through their phyllosphere, the leaf surface, and microbial colonization happens on the leaf surface just as it does in the rhizosphere, in the soil surface. So it's an investment by the plant because they want the ecosystem services of the organisms. It's an investment, and they're reaping huge rewards. They're, in essence, calling the specific communities of organisms that they want right around their root systems to get the specific nutrient cycling benefits, soil improving benefits, and immune system benefits that is provided by the organisms. So, in essence, they're building the castle wall around their root hairs. Castle wall of beneficial organisms There's a root hair. Well, it's not a root hair. It's an extremely rough depiction of a root hair. Secreting these exudates, the bacteria, fungi, protozoa, nematodes, macroarthropods, microarthropods are getting the nitrogenous compounds from the soil. They're cycling other nutrients right around the soil. They're taking the exudates, combining them together, building amino acids, building the basic blocks they need to grow and reproduce. And as a result, they're going to be outcompeting disease-causing organisms for water, for space, for nutrition. And so plant feeds bacteria and fungi. These microbes are gathering together the nutrients that they need in a very convenient place for the plants. It's a nice exchange. And so this is where I also want to emphasize the holistic nature of this process. And that's why I put this dimension into the talk, because when you start to get the big picture, you start to see how functionally interconnected all these parts are. Variations in the sun, the water, the weather, the wind, plant behavior, impacts the activity in the soil. So it's all one big system happening simultaneously. That's how nature designed it. And the more we've done to set succession back, by tilling, by putting chemicals on, the more simplified the system has become, and thus the more vulnerable it's become to disease, to extreme weather, to droughts, etc. Plants are more resilient when they have their wholeness intact. The relationship between plants and organisms isn't two separate systems. It's one system. And by simplifying the life forms in your soil, which may have happened before you even got there to work with it, and granted, most organic growers are helping the cause, depending on what it looks like, will depend on how strong that system is. Okay, so this is my friend Molly. She's running the Living Soil Lab now. This is some of our beautiful compost. But this is the living soil paradigm, what I've been talking about. This is where biology in the soil becomes more important than NPK fertilization. So, what is compost then? Good compost, like good soil, is a living ecosystem. Technically, it can be defined as oxidative decomposition of organic matter. The presence of air is absolutely necessary for the healthy process of decomposition and the full flowering of the beneficial organisms. So good compost will do basically two things. Inoculate the soil with a wide spectrum of microorganisms and provide food for the full array of organisms via organic matter and also other organisms, because they eat each other all the time. All of these notes, and there's some tables, all the tables so far are also online. The handout that I made available has stuff that we're going to get to, as well as my paper has everything that you see in here. So feel free to take notes if you want to, but you don't need to scribble stuff down. You can get access to it online. Okay. So knowing what we know about the living soil, here's some general guidelines for making good compost. The hallmark being active microbial biomass. That's how we assess good compost at the Living Soil Lab, and that's how good compost is defined. So you need the proper balance of air, minimum 6 parts per million oxygen, about 50–60% water content, enough heat, the result of the microbes doing their thing, and organic matter. Depending on what type of organic matter you put in and the ratios of the different types will depend on the outcome of your pile and the microbial profile of the pile. This data looks at the active bacterial and fungal biomass and protozoa numbers of 3 different substrates. Dry mulch, putrefied organic matter, which is nasty, stinky food scraps rotting in your backyard, and aerobic compost. Nice, loamy, delicious smelling stuff. You can see the first two, active bacteria, active fungi, almost doesn't exist. There's a ton of anaerobic bacteria in the putrefied organic matter. That's no bueno. Plants aren't going to like that. And same with the protozoa numbers in the putrefied organic matter. That's only ciliates, so that's an indicator that it has gone anaerobic if we couldn't tell by the nasty, stinky smell. And then when you look at aerobic compost numbers, much more active bacterial and fungal and protozoa. That means we're getting the full nutrient cycling, decomposing benefits, and that's what we're going to be inoculating the plants with when we put it on. Okay. Can you guys see that? Yeah, that's pretty clear. The color gradient. So this is a look at a windrow. They took a bucket loader and took a scoop out so you could see the inside. And on the top layer, that's okay. That's like normal color. As you start to get in, you start to see that white ring. That's an indicator of anaerobic conditions. This white stuff is actinobacteria, and it's kind of powder-like. Almost looks like mycelium, but it's not as pronounced. It's kind of thin. It won't hold together the particles like a nice mycelial fungal, wood chip pile would. And they thrive at 5.5 to 6.5 parts per million oxygen. So if you see that in the pile, you want to turn your pile and get some air in it. Otherwise, it'll go anaerobic. And this black middle has gone anaerobic. When you see black, think bad. Dark, rich brown, that's what's up. And you all know that, but the black stuff is a result of all the organic acids, toxic materials only produced in anaerobic conditions, formaldehyde, phenols, alcohol. Alcohol solubilizes membrane at 1 part per million. In anaerobic soils, you got that at 25 parts per million. That's going to turn your roots and all those beneficial organisms into jelly. So, the smell of vomit, rotting eggs, dead animals. If your compost smells like that, don't use it. Because that's what's in it. And also, it's going to be void of nutrients because the nutrients start to leave the pile once that anaerobic activity starts to occur because the nitrogen, the phosphorus, they off-gas. They change their form and they off-gas. So any benefits that you would have had, once it goes that anaerobic, isn't really worth using. Okay, and moisture. This is a basic squeeze test you can do. I got this on the worksheet too, or the handout too, so easy to remember. Reach into your pile at any given time, because you want to maintain that nice 50% moisture. That's the level that the organisms are going to do best in. They need that 50% water content to keep cycling, keep moving, keep having enough water to do what they do. So reach in the pile as far as you can, grab a handful of compost, and squeeze. If you don't get any drops out of that compost, you open your hand and it crumbles apart, you should add some water. It's about 20%, 20-30%. If you squeeze and you open your hands, and the compost is kind of got that shiny glaze of water, it holds together. You're close, about 40% moisture. If you squeeze and you get 1 or 2 drops, or water starts to pool between your knuckles, that's the sweet spot. That's 50, 60%. But anything more than that, more than 3 drops, is oversaturated with water. Don't water it. Cover it with a tarp. Make sure it doesn't get rained on. Because if you have that much water, the pile's going to go anaerobic, and we don't want that. So here's a quick look at a simple case study of good compost and not so good compost. These raspberry seedlings were put in potted compost mixes at the same time. One you can see is doing better than the other. So let's look at the one that's not doing so good. This was the bag compost that they bought at like a garden center. And they looked at it under the microscope. Really simple, really no food web present. Bacterial dominated, not many protozoa. They didn't see any fungi, they didn't see any nematodes. And I mean, you could see the plants, the leaves are dying, it's wilting. When they took it out of the pot, there was maybe not even a single new root. They saw one earthworm. The plant just never took off. They mixed this stuff with peat. So it was that bag compost with some peat. They tried it with that same compost, put some perlite in with the peat, try and improve the structure, see what would happen. They got only new root growth, was very thin. You didn't see any earthworms. And again, it didn't hold the root ball together. Just didn't have what it needed. So if you think you're using compost but without the soil food web, all you're really putting in there is organic matter at best. This was a homemade batch compost that they made. A lot higher protozoa numbers. Almost every slide they saw protozoa, bacteria, they saw some fungi. So more cycling of nutrients. The plant was a lot happier. Many new roots, finer ones and thicker ones. They saw earthworms in there. And you can see how that plant was doing. No groundbreaking science, but it's a basic example to illustrate the point. So once you get the good stuff, you don't need much of it. When you apply it, I mean, depending on your scenario, is it a market garden, is it a backyard garden, you got 50 acres, you're doing hay production, or you're doing whatever you're doing, it's going to vary how you're going to apply the compost because it's a precious resource. So I went into depth about different scenarios and how to apply application rates and stuff in my paper. So feel free to go online and check that out if you want to see. But in general, 1/4 inch to 1 inch layer can inoculate soil 6 to 12 inches deep. As long as you keep it moist, covered with some mulch. Don't put it out in the heat of the summer in a drought. So those organisms will really take off once you put them in there. And you know, if you got acres and acres of row crops, you're planting transplants, a water wheel transplanter, how nice would it be to pick out that seedling, inoculate it with all those organisms, and inoculate the whole bed. And it's a way to stretch out your compost and make good use of it because it is so precious. Liquid applications, extract, effective microbes. There's so many other different ways of people have used teas and herbal teas and stuff like that. How many people have heard of extract or aerated tea or have used it before? Okay, so you guys are familiar with this at least. Extract is, so here's the mesh bag that you're going to need. This is a big cone tank brewer. You could use a smaller 5-gallon bucket, but they have an aerated pump attached to this to make the tea. But basically, when you make the extract, you only need like a pound of compost for 5 gallons of water, so you can really stretch it. The rates vary whether you're making extract or tea. You need more compost per gallon making extract than you do for making tea, and I'll explain why in a second. But when you make extract, you put it into this bag, this mesh bag, put it into some fresh water. Make sure the water doesn't have chlorine or chloramine because that stuff's put in there to kill microbes. So if you can't get well water and all you have is the tap, you can use humic acid to mix in with the water to neutralize that chlorine and chloramine. If you have good compost, all you have to do is put it in a strainer, run some water through it. That passive leachate collected in a bowl is humic acid. So you can buy an implement from the garden store that you can attach to a hose, put that in there, and that'll be enough when you fill your bucket to make your tea. It'll neutralize that stuff so you can use that water. So you got the good water, you got the compost in the bag, and you just kind of agitate the bag for 30 seconds to a minute. The nutrients, the microbes will go into the water, and you can use that as a soil drench, put it around your trees, put it in a backpack sprayer, spray the leaves, spray your plants. There are a lot of different ways to do it. But if you want to make aerated tea, it's basically extract that you leave in the bucket or in the container for 24 to 36 hours. And you need an aerated pump to run constant air bubbles through it to maintain the aerobic conditions. The benefits of tea versus extract. Tea is better, I would say, for foliar applications. Because you get exponential growth of beneficial organism populations. Whereas extract, it's better at preserving the diversity of organisms because the aerated bubbles tend to break up the fungal strands, all that agitation overnight. So extract is a lot simpler, it doesn't take time, doesn't take the brewing motor or the monitoring. But if you want leaf colonization of the beneficials, tea is really great. Effective microbes are, some of you guys heard of effective microbes? Some people might have used it. It's like an anaerobic, beneficial bacterial soup. You order the culture, comes in, and it's one of the rare cases where it's a beneficial anaerobic inoculation in your water, but it lacks the diversity. Some people have really good success with it. Michael Phillips wrote The Holistic Orchard. He uses that effective microbes and like herbal teas, and he's gotten a lot of success out of it. He finds that the aerated tea and all that stuff is just too much to deal with and doesn't want to worry about it. He likes the results that he's getting. So you can try it out. I have more experience with extract and tea. They work great. You may need repeated applications to reestablish the biology, depending on where your soil's at or how your plant's doing. And my paper goes into more depth about the guidelines and timing for applying in different scenarios with an hour and a half session. I just can't get into it that deep because we're covering a lot, and hopefully it'll spark your interest in one way or another, and there's more information out there. Elaine Ingham's Compost Tea Brewer's Manual, 4th edition, has all that you would need to know to take this and run with it. And here's different ways you can direct inject it into the root system of a tree. I've seen that fully recover dying trees. Multiple injections, putting those beneficials right into the rhizosphere. You can spray it on crops. You can pull it behind in a pickup and spray your orchard trees. Whatever you're working with. Yes?
I'm just interested in the effective microbes. How is it if it's anaerobic? How could that be helpful if you want aerated biological communities at your root zone?
Because the main use of the effective microbes is foliar spray. So once you put it out on the foliar spray, it's not soaked in the soil. I'm not an expert in that. I haven't used it really at all. I've just learned about people who have used it, and they've had some success. But again, part of the point is broader diversity. The more you can, of the beneficials you can put on and put in, the more your plant's going to have to choose from when it secretes those exudates, the more options it's going to have to get the right communities. So effective microbes, they may give you some benefits, but getting an extract from a homemade compost that's full of the whole thing, that's really what you want. Can't beat it. So here's just another little example of some tea that was used on a football field. The dark strips were applied with compost. The light strips weren't. early September the year before, and this is what the field looked like after about a month of use. And that turf hadn't been able to establish. It was so compacted, and those root systems were just, they weren't working. A year later, late August, after a round of applications, that turf was able to really establish, and the plants were just, they were just healthier. The grass could really grow. And the biggest difference was the fungi in that soil as a result of the applications. Because bacterial populations are there, but when you get the whole web in, you get that aerating happening, you get the nutrient cycling happening, and all those nutrients that would be bound up in the bacteria get cycled through when they pass through the digestion of the third trophic level organisms, the protozoa, nematodes, etc. So this is the basic concept. Population, competition, disease suppression. And here are some numbers. This was a batch of tomato plants that got infected with a blight, and the researchers got them. They took one group, sprayed it with water, that was their control, all those plants died. That data is not shown. T number 1, you can see active bacteria, active fungi, very low. As a result, the colonization of the leaves with the beneficials was only about 27%. Only about 27% of the leaf was colonized by the beneficials. All those plants died of blight. T number 2, none died, and you can see the difference. Active microbial biomass colonizing, out-competing the blight, out-competing the disease, and those plants recovered. So, these are the basics. And again, the system dynamics should be clear. It's nature's design. It's enabling the system to function as a wholeness, as opposed to vulnerable, fragmented state. These systems have co-evolved over billions of years. So let's take it a little deeper. Let's talk about succession and evolution. Very rough depiction of succession. Bare rocks, parent material, photosynthetic bacteria breaking down, making topsoil, mosses, lichens, grasses, weeds start to grow, creating soil. Later, plants, shrubs, pioneer species, moving into the climax forest. This is what nature's doing all the time, just pushing for the next level of succession. And so down below, we have 3 different examples of farms. Some row crop vegetables, grazing animals. This is actually an apple orchard in New Jersey. And so your farm, every farm represents a stage of succession, and when you're farming a certain crop, you're holding that land at a certain stage of succession. And so it constantly wants to move on, but you're holding it at that level of succession. And just as the plants diversify and change, the corresponding soil ecology changes too. So when we look at soil biology and succession in terms of microbial populations, counts of bacteria and protozoa, lengths of fungal hyphae in a teaspoon of soil, we see bacterial numbers in the garden soil, 100 million to a billion, several yards of fungi, thousands of protozoa. Prairie soils, 100 million to a billion bacteria, tens to hundreds of yards of fungi, thousands of protozoa. Healthy forest, 100 million to a billion bacteria, 1 to 40 miles of mycelium in a teaspoon of soil, and hundreds of thousands of protozoa. So as you can see, the difference in fungal to bacterial ratios has everything to do with an increase in fungal biomass, because that range of bacteria tends to stay consistent. I mean, 100 million to a billion is a pretty wide fluctuation, but it's a consistent range throughout those systems. And there's a reason why these plants do this. Here's another way to look at it. Earlier successional plants, earlier successional stages have higher ratios of bacteria to fungi. They like a little bit more bacterial-dominated soil. Climax forests like very fungal-dominated soil, and then the spectrum in between likes a balance of fungi to bacteria. And there's specific reasons why these ratios exist. So here's a flow chart. It's another way to look at it. Early in succession, the niche is bacterial. Bacterial domination leads to microbial diversification, but then when you start to get early successional weeds, their niche is compacted soil. Their ecological niche is highly bacterial dominated soil, which leads to a high conversion of nitrogen, of ammonia, to nitrate. So high presence of nitrate as the predominant form of nitrogen, compacted soils, lack of oxygen, weed species thrive. As you go up, mid-successional grasses, vegetables, later successional grasses, row crops, like to see that fungal-bacterial ratio even out, and it has to do with the nutrient cycling dynamics of the organisms and what they make available to the plants in the soil. And then as you move up, shrubs, vines, bushes, deciduous trees. If you're doing like perennial polycultures or forest gardening, that forest, or just orchards, that forest edge ecology is the ecological niche for your 10 to 1 to 50 to 1 fungal to bacterial ratio. So if you're making compost and it's not fungal dominated, or if you're not introducing that fungal dominance to your soil, you're just not going to get as good of results. So this is another chart. Brassicas, annual flowers, early successional grasses, their fungal and bacterial preferences. Slightly bacterial dominated. As you go down, you can see how the ratios change. And these are general ratios, but it has to do with nutrients. It has to do with the relationships and how the dynamics change when the soil food web changes. So in nature, as in your farm, depending on the successional stage that your farm represents, and it might represent different stages or a spectrum of stages if you have some row crops, some trees, some grazing animals. Bacterial dominated soil converts ammonium to nitrate at a high rate. So nitrogen, four basic forms: ammonium, ammonia, nitrate, and nitrite. For our purposes, we're interested in ammonium and nitrate because those are the two forms of nitrogen that your plants prefer. Some plants prefer ammonium, some plants prefer nitrate. As you can see, early successional is nitrate, later successional plants, climax forests, old growth forests, ammonium. And then there's a spectrum in between. Of course, they want some nitrate, some ammonium. How do you know what your plant needs tomorrow at 3 p.m.? Does it want 3/4 nitrate and 1/4 ammonium? What does it need? Same with these other nutrients. Nitrogen is just one that's been studied a lot, so we have data. But these organisms are making the nutrients available to your plant in the form that it prefers, at the time that it needs it, and is holding those nutrients in the rhizosphere, in their bodies, and in stabilized, chelated forms, as opposed to soluble fertilizer forms, which are going to wash away. So you take this and start to customize your piles. This was the pile we built in Fairfield. Got really hot. And we built it way too big, so we had to turn it a bunch of times. It was a lot more work than we expected, but it turned out pretty good. So for our purposes, we can categorize the organic matter into 3 basic types. Brown, which has a carbon to nitrogen ratio of about 100 to 1 or greater. This would be the wood chips, the plant stalk material, all leaves, the materials that are high in lignin, high in cellulose, and that's what fungi feed on. Their enzymes and acids are designed to break this stuff down. So the more brown materials you put in your pile, the more fungal food you're going to be giving, the more fungal pile you're going to have. Green material is garden waste, hay, anything else cut while it was green that has higher sugar content and the carbon to nitrogen ratio around 30 to 1. High nitrogen, manures 10 to 1 or less. This gets the orgy going. This is the party food. This is the tequila of your compost pile. And it really just kickstarts the whole composting process. The more of this you put in, the hotter the pile is going to be. So making piles, best to scrounge around and get the materials that are readily available. And most of these materials are available, so where you shouldn't have to buy compost materials. Unless you want to buy bales of hay and stuff, which is really useful to use in the pile. Or bales of straw. And I said bales of hay, even though there's all those seeds in that hay. If you're doing a thermal pile, it's going to heat up and kill those weed seeds and pathogens. So there are 2 basic pile types, cold compost and hot compost. The hot compost is going to heat up 131 to 165 degrees. You get rapid decomposition. You get a more rapid finished product within, as little as 21 days if you want to work really hard and monitor it every day. But within a month or 2, you can have some really delicious finished compost, and it'll kill weed seeds and pathogens. Cold piles, also great. They stay below ambient temperature. It takes a longer time, 6 to 8 months or a year, but it doesn't kill weed seeds or pathogens. So you weigh your benefits based on your time, materials, amount of energy, management you have to deal with. So let's look at some thermal recipes. Depending on the percentage, like I said, of materials, you can select for a fungal-dominated or bacterial-dominated pile or a balanced pile. So depending on what plant range you're selecting for, you're growing for, should determine what type of pile you're building. And thermal recipes tend to have consistent 20% high nitrogen. That's going to heat your pile up pretty fast. And you can, depending on the potency of the manure that you use or the high nitrogen that you use, will determine how hot it gets, how many times you have to turn it. And I'm going to talk about turning and why that's important and how to do it. But for a bacterial-dominated pile, 50% green, 30% brown. Swap that for fungal-dominated, and even green and brown for a balanced pile. So if you're growing for mixed veggies, that balanced pile will give you that 1-to-1 fungal to bacterial ratio that your plants would naturally like to see. And for a hot pile, pretty much minimum of 1 cubic meter will give you the thermal mass to have that hot middle. You can set it up in an open heap, on a pallet, caged in, whatever works. Cover the pile with a tarp to control moisture. If you get a downpour of rain and it waterlogs your pile, it can ruin your pile, turn it anaerobic, and start all over again. That's not fun. So when you cover it though, don't seal it off. Don't put your compost in a plastic bag. But if you put the tarp over, leave the bottom air on and off. You don't have to cover it all day, every day. But if you know it's going to pour, put the tarp on there so that you can manage that moisture content. One tip when you're building the pile, water the materials as you build it. You have someone stand there with a hose, someone else is putting it on, or if you have a bucket loader putting it on. If you water it as you build it, I've had a lot more success getting those piles to get up to heat. Because if they're too dry, they're just not going to take off. Managing heat in the thermal pile is the most important thing because when you're managing heat, you're also managing air. Question?
Making these different types of piles, is it also correlated like vermicompost? You're using that in compost?
The percentages of green and brown, you mean?
Yeah, and can you alter the finished product of vermicompost the same way?
Yeah, so based on the foods that those worms like, it's mostly sugary bacterial foods, so that finished vermicompost will probably be bacterial dominated. But because of the way vermicompost is, I'm going to talk about it in a minute, it has the resources to balance out and become diversified over time. And if you have some fungal foods in there, like if you use a lot of cardboard or put leaves in there and stuff, it'll feed the fungi so that it can become fungal dominated over time. So starting temperature when you put it together will be ambient, but within a couple days, composting will begin. It'll rise up to the temperature, 131 degrees or more. That's the point where pathogens, weed seeds, and parasites begin to die of heat. You need 3 days at that minimum threshold for those pathogens and weed seeds to die, to be sure that they've died off. So 3 days before you turn it at that temperature range, 130 to 145. That's really the sweet spot. I mean, if your pile stays between 130 and 145, that would be awesome. That would be like the miracle low maintenance hot pile. But that rarely happens. Usually when it starts to heat up, it'll keep heating up. And it'll go up above 145, 155. Turn it within a day if it goes above 145. Turn it immediately if it goes above 160, because spontaneous combustion can and will happen at 180 degrees. And that's a fire that you can't really put out. Basically, the alcohols that are generated in that high heat uses up all the air, and it's explosive. So maintain 50 to 60% moisture content throughout the composting process. As you turn it, you can water it, but make sure you check that moisture content before you turn it so you know how much about you want to water it. And you're going to need a log thermometer. There's a company that sells a 4-foot long stainless steel thermometer. If you email me, I can give you that link. And you stick it in, you want to get averages from the middle of the pile, and that's how you'll know that middle ball is the hottest. That's how you can tell whether it's too hot, not hot enough, need to add more high nitrogen, need to add less chicken manure next time. Yeah.
I'm just wondering, what about like adding like ashes from your fireplace to your compost? Is that a good or bad thing to do, or?
I don't think it would make that much of a difference. I mean, if you're thinking about it in terms of what, like nutrients or organic matter?
You know, a way to get rid of it, but would it add anything to it? You know, I feel like I read somewhere that you can add it, but you can't reach a certain threshold or else toxicity or something along those lines. But
yeah, I mean, be careful what you burned.
Yeah, I'm just talking about wood from my wood-burning stove, but yeah, so
it probably wouldn't have that much available to feed those microbes, but I don't see the harm in adding it in there. If you want to just add some extra bulk, it's a way to recycle it.
Okay.
Yeah. Okay. So, turning the pile. Here's your pile, and for these purposes, we're separated into the top, the middle, and the bottom. The middle is the heat ball. The top and the bottom aren't going to aren't going to be that hot. So you want to make sure you cycle all the parts through the middle. So when you take the pile apart, take the top, put it to the side, take the middle, put it to the side, and then reconstruct your pile, making the top into the new bottom. And you might have, you know, a caged bin where you build your pile. So put the top in, becomes a new bottom, the bottom becomes the new middle, and that hot middle becomes the new top. And that way, every part of the pile cycles through the middle at least once. And that's important so that any pathogens, any weed seeds, gets a chance to be in that environment and neutralize before you put it out into your garden, or use it for extract or something like that. And this stuff is scalable. This guy, Jolly Farms out in Canada, they're doing it with machine windrow turners. So depending on your farm, depending on the resources you have, you might not do it at this scale, but you might want to collaborate with somebody who can manage that and make some really high quality stuff out of your animals' excrement, your field waste, or whatever you've got on your farm. And then cold recipes, definitely going to start with lower high nitrogen, definitely less than 10% high nitrogen. Variable green and brown, the same way you would build a hot pile with the green and brown, because that's just selecting for the microbial communities that you want. And you're going to want to balance air and water in the pile in the same way. Not necessarily with turning, because you might not have to turn it if it doesn't heat up. You shouldn't have to turn it. It's a cold pile. But you want to make sure you don't use too fine, like too much cut grass or something. You know what, mats gets anaerobic. So balance air and water. Make sure it doesn't get poured on. Make sure it doesn't dry out too much. And you can use that squeeze test. You can open it up from time to time and look at it, see how it's doing. So let's talk about earthworms for a second, because they're amazing. And most of you guys are probably familiar with the benefits of worm castings. And I'm not going to talk about how to set up a worm bin, because there are a lot of resources on that. But it can complement the larger system. It's so good for using as extracts, potting mix, because it's so nutrient rich and it's got a nice food web. When the organic matter passes through the digestive tract of the worm, worms cycle through tons of soil per acre. It goes through grinders. And the enzymes in the worm's digestive system is actually geared toward digesting the cytoplasm material of crushed organisms. It doesn't gain nutrition from organic matter. Microbes come in through the organic matter in the soil. It all gets crushed up. So that worm is grinding the organic matter, which is wonderful, because as it passes through the lower digestive tract of the worm, it gets colonized and inoculated with all those beneficials that live in worms' digestive tract, and leaves you with nutrient-rich, beneficial, microbial gold. And also pathogens, Salmonella, E. coli, that starts at the worm's mouth, does not come out the other end. So worm castings are great to use for extracts for that reason. I wouldn't want to use anything other than worm castings or hot compost for liquid applications, because anytime you put compost into water, you have that risk that you're taking. You know, if your pile has stuff in it that had pathogens in it, you use the manure that had pathogens in it, and it didn't get cycled through that heat or cycled through that worm's digestion, it could be problematic. Alright, how are we doing on time? Pretty good. So, I have time to talk about mulch for a second. Mainly as a complement to the composting system. Benefits of mulch are well known, but two in particular I want to talk about, two unique types of mulch are ramial wood chips. How many people have heard of ramial wood chips? A few of you. So it's the cuttings of fresh, like the tops of trees and bushes. It's ground up with the green leaves still on it, and the branches that are mulched to make ramial wood chips are about 1/4 inch to an inch. So they still have that nice cambium, fresh cambium layer behind the bark. So it makes a nice 30 to 170 to 1 mix of wood chips that's going to feed and support a nice range of microbial activity, great for trees and shrubs. If you can get that stuff, especially from power companies, if you can get it from them, it's really good stuff. And so it supports the deciduous environment as well as suited for orchard soil. It can be applied haphazardly in piles around trees or evenly across beds like other wood chip applications. Mushroom mulch. It's familiar with a lot of people around here because of mushroom growers. And it's the waste product of button mushroom farms. It's a good amendment for soil enhancement. And I know there's a little bit of controversial opinions about is it good, is it not good. Some people have had great success. Some people have had to kill them and burn their plants. And it depends on what stage you use it in. So it's the result of usually composted straw, chicken manure, sometimes horse manure, calcium, all those nutrients they put in, and then it's heated up to composting temperatures, and then cooled before inoculation. And the substrate becomes mulch after the mushrooms have flushed and been harvested, but there's still, because of the high nitrogen content in there, it's usually still hot when it gets delivered, and best to let it age. I mean, knowing what you know now, if you get some mushroom compost, assess it before you use it. Is it hot? Is it stinky? Does it smell like rotten eggs? I've definitely smelled mushroom mulch that smells like rotten eggs. On the other hand, there's a pile of mushroom mulch that's been sitting around for a while that at my friend's farm, and he's using it for as a top dressing for his plants. And he's got nothing but good results from using it. I would love to see it under the microscope. I haven't had a chance to look at it under the microscope, but it's generally got some nice fungal activity in there for obvious reasons, but it doesn't have the mycorrhizal fungi. Anytime you process the compost through the heat, it kills the mycorrhizal spores. So while you will be getting really nice fungal activity in your pile, you might want to add some mycorrhizal inoculants to get those benefits. Mycorrhizal fungi are some of the best root mutualists. They transfer nutrients and water between plants. They've been shown to support dying trees and sick trees by transporting nutrients from one tree to the other. They're the web that connect plants underground. So it's generally not used as a soil by itself because of the nutrient imbalance. Any questions up to this point? Cool. Yeah.
You mentioned a handout that you had. I'm sorry, I haven't gotten it. Is it something that
I think
should have picked up?
they have. I gave it to the guys at NOFA, the ladies at NOFA, and they should have it available for printout. That's what they told me. It's on the web. It's on the web. It's also on my website too. Yes.
Why is the ramial wood chips require deciduous trees rather than softwood? What's the difference?
It's a good question. Mainly because in orchard soils, you do more deciduous environment. So, as far as the microbiology is concerned, I'm not really sure. It's a good question.
Yeah, I mean, I know that Michael Phillips talks about it in his book, and I'm trying to wrap my head around it. Haven't quite got there yet.
Yeah. It may be due. The only thing I can think of would be the bacterial food that it provides, where those other systems might not want to have bacterial elements. One of the things I forgot to mention in the fungi bacterial spectrum and the nutrient cycle, fungal dominated soils suppress the nitrifying activity of the bacteria, so that conversion from ammonium to nitrate is a result of nitrifying bacteria. And fungi secrete an enzyme that inhibits that activity. So the trees that send out exudates selecting for higher concentrations of fungi, they're feeding them through the materials that they put down, they're adjusting that nutrient cycle that way. So fascinating science. So there is a link to my website. I have the full paper PDF there, which has some resources in the bibliography. And my link is also on the handout. You got it? Yeah.
I was curious if, at the micro level, there's any concern about bringing in invasive or non-native microbes, like, and, getting compost from, getting mushroom compost from Pennsylvania, for instance, and bringing that into your farm, that's bad for your local soil ecology or community?
No, it's not a problem. The more diversity, the better. Remember, the plants can manage those microbial communities. So if there's stuff there that they don't want, but they have the spectrum of organisms, they're going to be just fine. My understanding is the more diversity, the better. Yeah.
Are there any weed seeds or weed species in particular that you know that are more resistant to cooking out the piles, or am I just not mixing my piles well enough? Because land squirter in particular, I have had problems with it cropping up out of what have been really hot, well-managed piles.
Maybe that they didn't spend enough time, there might be parts of the pile that didn't cycle through that hot middle long enough.
But in your experience, the heat, time and temperature should kill off any of the weeds.
Yeah. All right, so we're going to have time for more questions, but let me just finish up. Here's the page 1 of the handout. It's got the materials list, some recipes, reading moisture content, my information, as well as these successional charts for you to have at a glance. So this is the art and science of compost. Talked a lot of science this morning, and let's talk a little art. Organic gardening is complex and simple. Blend of good science, fact, experience, intuition, experiments, play, speculation. The art of composting is the art of transformation, the art of succession and evolution. It's an ongoing process. If you're willing, you can begin to tune yourself to that process and become sensitive to the relationships between the plants and the living soil. The best way to work with this stuff, as with plants in general, is to establish a dialogue and get a feeling for the aliveness of the process. So please remember that the science of compost, the science of the soil food web, is all metaphor. Cation exchange capacity, we didn't have time to talk about that. Carbon nitrogen ratios, nutrient cycle, they're all metaphors. The reality of the soil food web is beyond our ability to express in words, which is why plants don't grow in books. The intelligence of the soil food web goes beyond beyond our maps and diagrams, cannot be explained by our facts and figures. The reality of life, composting, farming, is not easily predictable. The paradigm of living soil acknowledges the vital interconnectedness between microbes and plants, air and water, sun and people. What we do to the web, we do to ourselves. The earth is not a machine. And even the tiniest changes in one place can change everything in ways we wouldn't expect. So we began the session today by envisioning our current farm and garden and seeing its fullest potential in our dreams. Now, I want you to close your eyes once more, please. Take a deep breath. See if your vision has changed. See your farm once again. Look around. How does it feel? I hope you can see how the soil food web and composting can support you in your mission to have the most abundant, natural farm. But I also hope that through the art of composting, you can stay connected to an even bigger picture. You can open your eyes if you want to. So my passion for the soil food web is a passion for life itself. I never thought I would geek out on applied soil microbiology as much as I have, but it's great. When it's understood that one loses joy and happiness in an attempt to possess them, then the essence of natural farming will be realized. The ultimate goal of farming is not the growing of crops, but the cultivation and perfection of human beings. I understand that if you're committed enough to sought out this information, you already consider yourself a natural farmer or gardener. Again, thank you so much for coming. As you take the information, compost pile recipes, materials list, and instructions, and prepare to apply it to your current operations, do your best to carry yourself more as a midwife than as a chef. Realize that with every compost pile you make, you're giving birth to an entire community of new life forms, and your farm will be a stronger community because of it. Like any good community organizer, which you are, you need patience, flexibility, problem solving, all those good things, which you already know. So engage in the process of growing more abundant and healthy plants so that you may become more healthy and abundant in spirit. Farm so that you might create more harmony on earth. Your job isn't easy, and you know it. And it goes on forever. Thank you. About 10 minutes. Yes.
I'm having a lot of trouble with my compost pile with the leaves I added to it. They're really matting down and creating it hard to get water to it. And, I was wondering what's a good way to go about changing that.
Cut it in half with wood chips. Something with a little bit more. And the wood chips, best if they're an inch, no more than 2, the way that they'll get broken down, but yeah, that'll add some more air pockets.
Will that add a lot of time though to the decomposition too? Or?
If they're thick chips, but you might get some chips with still, in your finished pile, but that'll just add more food as you put it down. So as the process goes on, there's the immobilization phase and the mineralization phase in the compost pile. So when you put the materials on, especially the more complex compounds, the wood chips and woodier stuff, it's going to take a little more cycling for the nutrients that get bound up in the bacteria and fungi to get released in plant available form. And that's the product of the protozoa and the nematodes, and that's why it's important to have them in there. Because if you don't have them and you're adding mulch and stuff like that, the bacteria and fungi are going to tie up the nutrients. So as long as you have the web, they'll cycle through, and it'll just be like fertilizer basically over time.
So are the nematodes and the protozoa basically like feeding on the fungi and bacteria? Is that how they're making it available to the plants?
Exactly. Their excrement is plant available chelated nutrients, soil stable.
I was curious about, I know that brassicas are more bacterial instead than fungal.
Yeah.
So I've been using some like mycorrhizal applications when I make potting mix, and I don't do it with brassicas, but if I'm applying a compost or a compost tea, would it be best to use something that's even less than balanced, or is balanced okay for brassica plants?
If you're making a compost tea for brassicas, when you make the extract, you're going to add some food in there, and there's bacterial food and there's fungal food. So if you add like molasses or something, that'll just select for more bacterial dominance in that tea, which those brassicas would probably like a little bit more. Yeah. Yeah.
Do you add stuff like biochar or like rock dust in your compost? And when should you add those?
It's not necessary.
No.
It's not needed at all. It's a misconception that you need minerals in the soil. There's even calcium, even stuff like phosphorus. It's all there. Like I could send you charts. You could email me. I could give you charts that show the available. Okay, so there's the extractable and the available nutrients, and then the total nutrients, the total minerals, and it's the microbes that you need more than the minerals, because if you have the microbes, they're going to make all those minerals mobile and plant available over time. So even if I would say adding biochar, adding rock dust would inhibit plant growth more than it's going to help it with the microbes. If you don't believe me, I could send you some information.
I did a soil test for the local labs, and I was deficient in stuff like molybdenum, sulfur, a lot. So I figured.
Right, but they're not testing what's in the bodies of your bacteria and fungi.
Well, I would think the rock dust would be like there for them to break it down for nutrient.
It's already there. Even the molybdenum, it's all there. It's just that they're bound up. They're not plant available. And if you have the soil food web in place, they're going to make it available. That's their job. That's what they're doing. That's what they're doing at the bedrock. They're making all this stuff available. But when it gets to your plant roots, if you put it all down, I mean, what are soluble minerals? They're salts. So they're gonna kill the organisms more than anything. And that's going to reduce the ability of the soil to mobilize those nutrients for your plants.
I've had a lot of problem with mildew in my garden, and there's a lot of vegetables that I had to stop growing because of that. The first year was great; I didn't have any problems, and then in succeeding years, it got worse and worse. If I'm doing this right in adding the good quality compost to my garden, is it going to solve that problem eventually?
The mildew?
Yeah.
Yeah, it will definitely solve that problem. And if you're having problems on the leaf surface, make some extract.
Mm-hmm.
Colonize the leaf surfaces of your plants. Cool. Anything else?
What's your thought on hugelculture?
What was that?
The hugelculture? Since the German
Mm-hmm.
What's your thoughts on that?
Awesome. Great for perennial plants. really great for perennial plants, but you need the wood.
After this past winter.
Yeah, and especially after the storm, there's a lot of wood. Yeah, over time, that's just going to feed those fungal populations. It allows space and air in the soil. So even if you have thick hugelkultur beds, all my experience is that they're great for the long-term buildup of humus and organic matter and supporting those fungal populations. Might not be so good for like brassicas, as you say, or earlier successional plants, but yeah.
Can you briefly explain what you guys are talking about?
Hugelkultur is, I guess, developed by Seth Holzer, and it's a method of building raised beds by digging down and adding branches and logs, and then covering that with soil. So the structure of that raised bed is thick branches and logs, and you grow your plants directly into that. So over time, it all decomposes, and it's like a feedstock for the organisms. And another thing for like wood chips and in your compost, like certain pines, juglans, other things like that will have terpenes and phenols that off-gas and might inhibit plant growth to some degree, but like pine chips or pine needles, if you give it some time to age, that off-gassing will happen, especially if you like turn that chip pile once. You'll be able to tell when it's not so strong that they shouldn't be a problem. Yes, sir.
Some of the counties have programs where they supply compost, by the truckload or yard load. Do you have any experience with, I think they make them out of leaves mostly, whether that's good or bad or tested or not, or is it individual? Are you familiar with those programs?
Are you thinking mostly municipal waste and leaf mulch and stuff like that?
Yes, exactly.
It really depends.
Depends.
Standard ecological answer.
Yeah. Right.
But I would have to look under a microscope. In general, that leaf mulch would be more fungal because those dead, dry leaves are fungal food. Good organic matter, but again, with municipal compost, you gotta be careful with what they're putting in there. It might be toxic heavy metals. And the organisms can decompose toxic material. Paul Stamets has a lot of research about specific types of fungi and adding it to decompose different toxic materials. But a lot of that stuff just doesn't have the diversity, the microbial diversity, and a lot of it too has gone anaerobic. And so it might look like rich and dark and great, but it's a lot of those nutrients get burned up, and the microbes haven't really grown properly.
So just good for organic matter, maybe more than compost.
If anything, yeah.
Do you think they might do testing on it at the local counties? Yeah, it's a question I could ask them.
Yeah, you could ask them. I doubt they're doing direct microscopy and counting, because that's what we used. That's what Elaine Ingham teaches for counting and assessing the microbial communities. Because if you just do a plate count and say, okay, trying to grow a certain type to see if it's there, that's not really going to help you see what's available to your plants in the medium that you're going to be putting on. That's another session in and of itself. But, yeah.
Like a drip tape irrigation system, I was interpreting what you said as the extracting better and having because it doesn't break up fungal hyphae and a seed more for foliar applications.
Yeah, you can use extract in foliar applications too. It'll still be good, but the benefit of tea and the foliar applications has to do with the time that it's had to cultivate the higher populations of organisms that are going to be more successful at colonizing the leaf surface, the entire leaf surface of the plant, and the stems and everything like that.
Okay, at our farm we were doing a feed, and for a few days in oxygenation, and then running it through a hose. Would you recommend, therefore, to extract something and do it one day? Basically, you run it through the you know, you put the raw material in, take the water out, and then plant, you know, water your plant, right?
Mm-hmm. Yeah, it's best to use the stuff within a couple hours.
Okay.
If you let it sit around too long, stagnant water is inevitably going to go anaerobic. All right.
Did you discuss with green tea, making water with like stinging nettles, fresh cut?
Yeah.
That's not things.
I don't have much experience with that, but I know Michael Phillips has different recipes and ways that he does it. I mean, he talks about like protozoa infusion, where you let like stuff like these herbs sit in water overnight and they go anaerobic. I would want to look at them under the microscope before I put them on my plants. But I'm an herbal medicine practitioner, and I know the benefit of these plants are for the plants as much as they are for us too. But those would be more nutritional benefits than microbial.
Can you tell us about your, is this your company? Is it a business or an educational thing or what?
Both. Yeah, it's actually it's out in Fairfield, Iowa, and I'm just affiliated with them now because I relocated here to pursue a graduate degree. But the Living Soil Lab is the microscopy to assess the soils and the compost and tea that they make. So it's talk more about the company there, but it's out there. All right, that's time. Thanks everybody.